Hi, my name is Peta Bradbury, and today I'm going to talk to you about some of the work that I did before I left Sydney, where I was at the Woolcock Institute of Medical Research, which is a respiratory-based medical institute. So the data that I'm going to present is focuses around the impact that space may have on tissue function, and this led us to establish an emerging role for gravity or microgravity, and how this plays into regulating the structure and function of tissue epithelia. So the tissue that we chose to work on was the nasal cavity. And that's because the nasal cavity has this really interesting feature known as the air-cell interface. So what I mean by that is when you inhale through your nasal passages, the air will flow over and pass over those cells, like exposed cells, for lack of a better word, really. And this is a really interesting phenomena, and it helps promote the differentiation of the epithelial cells, but also it helps promote and establish the integrity of that epithelial layer. So if we look at the epithelial cells down here, what we'll notice is that there's three main types of cells that you'll find in a nasal epithelia: the basal cell, which is essentially almost like your progenitor cell. It's the beginning one that kind of everything establishes from. So from the basal cell, you can have a ciliated cell. So ciliated cells obviously produce the cilia that are important in creating that movement to move the mucus out of the airways, but also goblet cells, which are important for those, for secreting the mucus essentially, and act as that, the buffer between the air and the cells. So this air cell interface kind of promotes a certain cell structure, also gives rise to cell function via that differentiation, and then the integrity of that epithelial layer is kept via these things called as cell junctions. So tight junctions are part of the cell junction family, and all junctions are formed between the cells. Essentially, they're like a little key that kind of locks the two cells together. So this forms the structure and gives integrity to the structure itself, to the epithelial layer. So we wanted to look at the way that gravity might influence or regulate these particular functions of the nasal epithelia. So the first thing that we did was really decide on a way to simulate that microgravity, and we teamed up with Xerxia. And we used their random positioning machine. So what you'll see in this movie here is essentially the machine in motion. So it rotates around a double axis, and giving three-dimensional rotation, providing a random change in the orientation relative to the gravity factor. I'm not a physicist, so I can't answer any questions about this, but if you do have questions, please ask Josh. I just supplied the biology. So moving on, we subjected our cells to microgravity in order to determine their cell structure. So we used the RPMI 2650 cell lines. This is a nasal cell line. First thing that we did was just stain these cells for actin. So actin's a cytoskeletal marker, quite an important cytoskeletal marker. So what we can see here in the red is actin at its highest intensities, essentially, which is what you're seeing here. And actin gives rise to cellular structure, but also cellular shape, and is actually quite an important component of the cytoskeletal system. In Dappy in blue here, we have the nucleus, and then your merged images will always be on the right. So these are what the RPMI 2650 cell line looks like under control conditions. They're quite aggregated and quite bunched up almost. When we subjected these cells to microgravity for 24 hours, just 24 hours, and we fixed them and then we stained them with the same stains, what we found was that not only were there less cells, which is not a story I want to talk about today, but we have definitely looked into this, and that's another story. But today I want to talk specifically about the shape of the cells. So what we found is that the cells that were exposed to microgravity started showing these lamella. And very nice leading edges almost, suggesting that there was a complete rearrangement of the actin cytoskeleton. These cells were now no longer wanting to be as aggregated, but not only that, they were now bigger and longer. And so we quantified these changes, and indeed what we found was that the exposure to microgravity did significantly increase cell area. We then moved on and looked at aspect ratio. So aspect ratio is a measure of the length of the longest chord length divided by the shortest chord length. Essentially, it's a length by breadth analysis. And what we found was that when we exposed those cells to microgravity, they did end up generating a more elongated phenotype. And finally, we looked at the actin fluorescence intensity per cell. And what we found was that there was a small yet significant increase in the amount of actin present in the cells, which kind of paired very nicely with what we were seeing with our rearrangement of the actin cytoskeleton. So now that we had determined that microgravity did play a role in changing, or at least mediating changes in the cell structure of these cells, we moved on to look at differentiation and tight junction functionality in particular. The way that we do this in the respiratory field is we expose these cells to, well, first thing we want to do is really recapitulate that air-cell interface that I spoke to you about earlier. And the way that we do this is by creating what's known as an air-liquid interface. And we use these things known as transwell inserts, which are here in the grey. And what's important about these is that at the base of this, it's a semi-permeable support membrane, and that comes in really becomes important in the next couple of seconds. So the first thing that we do is those cells were exposed to microgravity, and then we placed them in the apical chamber, so the top chamber of that transwell, and then filled the basolateral, the bottom compartment of that insert with media. After 2 days of a growth in this condition, the media was removed and the cells are exposed to air. They're allowed to grow throughout this model, throughout the next 21 days, in fact. And the reason that they're able to grow is because of this semi-permeable membrane. So this membrane here allows the transfer of nutrients and growth supports throughout the basal compartment, allowing these cells to grow for 21 days. So if we take a closer look at this cell compartment, what I want to show you is kind of what happens throughout these 21 days. So in order to fully recapitulate the air-cell interface that we see in vivo in the nose, what we want to see throughout that 21-day period is the differentiation of those epithelial cells to goblet cells. So we want to see the differentiation of cells to the mucus-producing cells, sorry. But more importantly, we want to see tight junction formation. So we want to see that we have an epithelial layer that is full of tight junctions to allow that structural support that we spoke about earlier to give structural support to that cell layer in the apical chamber. So to give you an idea of what this looks like in real life, essentially, so this is an image from a paper from a German group that used the RPMI cell line as well. And this is a H&E stain of a transverse section of the ALI culture. And what you'll see down here is this semi-permeable support membrane that I was been speaking about earlier. So this allows the transfer of those nutrients to the growing cell layer. So you'll notice that the cell layer is actually quite deep. So in particular, it's known that the RPMI cells produce a cell layer of around 14 to 20 cells in that 21-day period. So throughout this entire cell layer here, you'll notice, well, you would hope that you would have tight junction formation to fully recapitulate that epithelial layer. But not only that, you want to have mucus secretion to prove that you have differentiation of those cells to now goblet cells and therefore mucus producing cells. So the first thing that we did with our model was actually look at the live dead staining of these ALI cultures, because remember, they'd been in culture for essentially 23 days all up, and they were, some of them, well, the microgravity cells were exposed to microgravity for 24 hours and then placed in ALI. So we wanted to see if that when we were doing our experiments, they were actually live cells there. So these are epifluorescent images of live and dead cells. And what we found here was that whilst we're unable to quantify them numerically, because these are just epifluorescent images, they are not confocal slices, we can see that there's a proportionate number of live and dead cells given the two conditions. So this made us comfortable to move forward with our experiments. So the first thing that we looked at was mucus secretion as a surrogate marker of differentiation. And that's because only goblet cells will secrete mucus. So we looked at the staining for mucus to determine essentially differentiation. So we use a stain known as Austrian blue, and Austrian blue stains the peptidoglycans present in mucus, and it gives you this blue intensity. So the idea is that the higher ratio of blue, or the more intense blue that you see, would be an indicator of greater differentiation, i.e. greater mucus secretion. And then if you had a lower intensity of blue, it would suggest lower differentiation. And when we quantified this using the ratio of blue over red, green, blue, which is a common marker or a common equation that we use in the field, we found that there was no significant difference between the two conditions, suggesting that microgravity had not actually influenced differentiation in these cells, which for us was an interesting finding and a good finding. And now we went on to look at the tight junction functionality. So just to give you an idea again, just to reiterate, what we wanted to look at were the integrity and functionality of tight junctions. So tight junctions are shown here in the green, and then in purple here you have the two walls of two separate cells, and then the tight junction almost forms a bridge between those cells that locks them into place. So here we have the apical surface, reminiscent of the air surface here, but then the basolateral side here, reminiscent of your basolateral compartment here. So these tight junctions here, the green complexes, they're made up of quite a lot of different proteins, to be honest. And what we typically look at when we stain for these cells are two proteins. One, E-cadherin, because E-cadherin is very important in the formation of junctions, all junctions, cell-cell junctions and tight junctions. But also we look at ZO1, because ZO1 is an important marker of tight junction transport. So it kind of gives functionality to these tight junctions. So if we have a solute that is in the apical layer of or on the apical surface of your epithelial layer, they can pass through these cells, then in between the cells via this paracellular space and enter the basolateral compartment, which means we can measure things like junctional integrity by measuring the change in electrical resistance from the apical down to the basolateral, but we can also then use specific chemicals to then measure the rate of flux of these chemicals or solutes through that apical and apical surface down to the basolateral. So this is exactly what we did. We measured junctional integrity first by using the ESIS, which measures the change in electrical resistance essentially over time of these ALI cultures. So here we have normalized resistance, and then this is over a 24-hour period. And in blue, we have the control. And what we can see is when we look at the red microgravity line, you see this drop in normalized resistance, suggesting that perhaps those tight junctions are not as integrous or are not fully formed as we would expect in the control setting. We then moved on and looked at paracellular transport. So we used a marker of paracellular permeability. So we used essentially what I'm saying is we used a chemical known to only be transported via the paracellular route. And when we measure this, we quantify it using a PAP, and we measure that in centimeters per second. So how much of the solute is transported through that tight junction, through those tight junctions via the paracellular route. And what we found was that when we have cells exposed to just 24 hours of microgravity, we have a significant increase in our PAP, which actually suggests that these tight junctions are not functioning as well as the controls, because they're allowing that solute to pass much quicker through the epithelial layer, suggesting that then functionality is not is somewhat impaired by the exposure to microgravity. This got us a little bit intrigued. So what we then went on and did was look at those two markers that I told you about earlier, ZO1 and E-cadherin. So we have ZO1 here in green, and then E-cadherin here in red. And then again, our merged image on the right. And what we see with ZO1 and ECAD for our control cells is a very nice peripheral staining around the cell for both the ECAD and the ZO1, which is what we would expect when we're looking at confocal Z slices. We want to just see this peripheral localization of the peripheral localization of the two proteins, suggesting that we have tight junctions forming between those cell-cell neighbors. When we looked at the microgravity exposed cells, what we found was that there was absolutely no co-localization or subcellular localization to the periphery. So both ECAD and ZO1 were found to be dispersed throughout the cell. There was no targeting to tight junctions. They didn't really co-localize together in terms of their tight junction structure. So this kind of made sense then. We've got lower integrity of the tight junctions for the microgravity cells. We also have a higher PAP, indicating that the tight junctions are not functional. And then our IF staining has proved that we have no localization of these two tight junction markers to the tight junctions, suggesting that these structures are just not formed post microgravity. So this is where I'm going to leave today, but this is really exciting for us because it really does open up a whole area of new medicine, essentially, because tight junctions are very important in not only nasal epithelia, but they're also really important in regulating epithelia of the lumen, of any lumen. So colon, the stomach, all of these things. So if we can find a way to manipulate and increase the permeability of these tight junctions in the luminal area, we might be able to deliver targeted drugs to these areas. Now that's really exciting for things like cancer therapeutics, but it's also really an important field for drug transport. So there's two kinds of like drug formulation and then the biology behind it. So it's really, really exciting. So we're working on the mechanism at the moment. So with that, I would like to acknowledge everybody that has been a part of this work, namely Katerina. So Katerina was a phenomenal intern that I had who came across from Italy to do her her bachelor's. So they did like a 4-month exchange program with my team. She'd never been in a lab before, and she generated this work and so much more of this work. She was phenomenal. YY is my boss, who I think thought I was a bit crazy when I brought this project to her, but she fully supported me, and I am so ever grateful. Aylin and Zara have been an enormous help in just finishing off some of the projects, and of course Josh and his team for just being amazing people. So with that, if you have any further questions or have any questions or comments or anything like that at all, please feel free to email me. I will absolutely reply to you. So with that, thank you for your attention and good night. Bye-bye.